Hello, good afternoon, everyone. We'd like to welcome you to our summer research seminar series here at HOPP. And for those that are joining for the first time, my name is Vicki Bowden and I'm a project manager here. Before we begin, and I'll introduce our speaker. A couple of reminders. We do have a Q&A feature. Anyone that has any questions, feel free to submit them there. And at the end of the session, myself and Dr. Sen will address them. We also we'll send you all an attendee survey for you to share your feedback based on how you found this seminar. Also, we will be emailing you all tomorrow regarding our registration information for our PI Spotlight session. So please stay tuned for that email as well. And if you haven't viewed past recordings, they are up and available for viewing on our HOPP Summer Student Program YouTube page. We welcome you all to visit and as well subscribe. Today's session is Animal Use in Cancer Research, and we have Dr. Triparna Sen, who will be speaking. She is an assistant attending at the Department of Medicine here at MSK and has a broad background in translational oncology research with specific training in lung cancer, DNA damage, response, immuno-oncology, and drug development. She has identified that the DNA damage response proteins are viable therapeutic targets for small cell lung cancer treatment. She was the first to demonstrate that targeting DNA damage response promotes anti-tumor immunity through activation of innate immune signaling in small cell lung cancer. This work led to a preclinical platform to discover therapeutic strategies to reactivate anti-tumor immunity in small cell lung cancer. The long-term goal of her research is to translate new mechanism-driven insights of cancer biology and immune oncology into bringing novel therapies to the clinic for lung cancer patients. Again, we welcome Dr. Sen.
Thank you and welcome everyone who are attending this seminar series. I'll share my screen now. So as Vicki mentioned, today I'll be talking about animal models and cancer research. And animal models are really key for doing most of the research that we do in the lab right now. But before I begin, I would just like to commend everyone for doing so well. We live in very interesting times, and I hope everyone is maintaining social distancing, staying safe, wearing a mask, and seeking help whenever they want to and reaching out to people. You are welcome to email me, send a message on Twitter, and contact me at any given point in time to ask questions about your career choices regarding this seminar or anything to do broadly with oncology. I'm more than happy to answer your questions. You already know about this amazing seminar series that Memorial Sloan Kettering is hosting, but I just want to point out that there are great seminars coming up in the coming weeks, and I would recommend that you all register for these seminars. So with that, I would go into the topic. So what is an animal model? And why do we need models? So there are many kinds of models through which we study cancer research. You have your in vitro or the cell line models, you have computer models, you have mathematical models, or today's topic, which is animal models. The goal of any model is to recapitulate what happens to the disease in the human. Animal models may be analogous processes that are processes that have similar function, or homologous processes that share similar embryonic origin or genetic sequences. You have to be very careful when you choose your animal models, and there are some models are better for a certain type of disease than the other, and I have just mentioned a few here. Like Drosophila is used very much for studying developmental biology because they're small, they have short generation time, but the disadvantage is they're not vertebrates. fish. It is also used a lot in developmental research. And again, it has a short generation time. It has clear embryos, but they are not mammals. The animal that we use mostly for cancer research is mice. They're mammals, they're relatively small, they have a shorter lifespan, but again, they come with the disadvantages that they are not primates. But having said that, today, majority, almost the entire talk would be dedicated to having mouse models in cancer research, because that's a tool that we use most frequently. But rats are also used. They are bigger than mice, but again, they are used for cancer research nevertheless. People use dogs for studies like bone marrow transplantation, but they're costly. There are ethical issues. The bigger your animal model gets, there are more ethical issues connected to that. And then again, people use non-human primates, but again, there are ethical issues connected to using these higher models. So the uses of mouse models in cancer and why do we use it is to study cancer gene function. That is to study a certain gene and what it does in the human disease. That's why mouse models are used. It's a source of genetically defined cells of many types that you can isolate cell lines from different tissues, and you can use those to create or generate tumors in mice. To understand the biology of specific diseases, that is, you can engineer mice to mimic a certain type of cancer. In my case, I have studied lung cancer extensively, so I have used cell lines and models to mimic what happens in a human lung cancer patient. To understand the role of environment in disease and its interaction with genetics, and I'll go a little bit of that when I talk about chemical-induced mouse models. I think the best part is you can study pharmacology. You can study how drugs, interaction of drugs with cancer with animal models. I think that's where it has found its most use other than doing the genetic studies. And I'll go into much detail about that. And also doing efficacy screens. Like when you do clinical trials with your drugs, you would likely want to test the drug preclinically in an animal model. And we do extensive preclinical studies in mice in the lab before we take a drug to the clinic. And you can better interpret results. Like when you're doing a clinical trial, you might identify biomarkers or other genes of interest or other drug interactions, which you can then again take back to the lab and do co-clinical trials or post-clinical trial studies in a mouse model. The mouse model has completely revolutionized, and there has been an explosion of how much mouse model is being used in cancer research right now. This was a review that was published. What you can appreciate here is that pre-2000, you had a handful of mouse models like knockouts regulated by cDNAs or with chemical mutagenesis. But post-2011, you have these very sophisticated newer models that have now been introduced, and the list has only grown bigger and bigger with time. Like every year you get new mouse models with the new genetic manipulations. And now with CRISPR, it's a completely new field where genetic editing is the next generation for creating mouse models. So what you can appreciate is that mouse models have completely changed how we do cancer research right now. So what is your ideal mouse model of sporadic cancer? So cancer arises from a sequential accumulation of changes. It could be genetic or epigenetic that drive the conversion of a normal cell into a malignant derivative. And my previous speakers in this seminar series have spoken to you about the disease, about how the progression happens. So it ideally happens over time. So you would want to study or mimic the genetics and the cellular pathobiology of cancer through your mouse model. You want to look at mutation in a single cell level and also its interactions with the normal microenvironment. Your mouse model should ideally have a mutation that you can control spatially and temporally. What I mean is that you should be able to decide where to put your mutation in and when to put your mutation in. So that's what I mean by spatially and temporally. And multiple mutations, so because cancer is a complex disease and almost always you would have more than one mutation that causes a cancer. So your ideal mouse model is the one where you can introduce multiple mutations throughout the course of your study. And tumors can be tracked over time in response to treatment. So you should be able to have a model where you can see how the tumor growth is happening and whether your drugs are being effective or not. So when you're designing your model, these are the things that you have to keep in mind when you are doing cancer research. Today, I'll be talking about spontaneous mouse model, which can be chemically induced or viral induced. I'll be talking about xenografts, which can be derived from cell lines or patient-derived xenografts, and a newer technology where we can isolate circulating tumor cells from blood of patients and make mouse models from there. And I'll be going deeper into genetically engineered mouse models because, as I said, it has completely revolutionized of how we do cancer research. I'll be going into non-germline and germline GEMMS, how you can make a constitutive versus conditional mouse model, and the methods of germline modeling. So spontaneous cancer models, they are of specific strains that are at high risk of developing specific tumors. Now, like the New Zealand black mouse, it has a spontaneous development of chronic lymphocytic leukemia. So this model has been used extensively to study this particular disease, where a germline mutation in the same microRNA as a subtype of human CLL can be studied. The advantages of using this particular model is that it is a homogeneous system. It can be genetically manipulated. As I said, you can introduce this mutation and hence that leads to that disease. It has transferred immune competent secondary recipients. You can make these mutations in an immunocompetent mouse model, and thus that helps you to see the interactions with the tumor microenvironment. But the disadvantages of having this spontaneous cancer mouse model is that it has a variable latency and penetrance. So you may not be able to introduce these mutations in all cells that you would want. And in many cases, the genetic etiology is not known. So there are advantages and disadvantages of every mouse model. And you will appreciate that as I go through my presentation, is that no model is really perfect. You just have to choose the best that you would need for your research. The next is a chemical-induced carcinogenesis model, and there are several. I would just talk about a few. A number of chemical and radiation carcinogenesis can introduce the insurgence of spontaneous tumors. And in many cases, chemically induced carcinogens, they very faithfully recapitulate the genetic makeup of the disease. And this is just one example of that. This was published from the Barbacid lab in Nature in 1985, where he showed that N-nitroso-methylurea induced tumors that acquire HRAS mutation during carcinogenesis. Again, the advantages are the in vivo selection for genetic events that are very relevant to this particular type of cancer. These models are relatively cheap and easier to use. They can effectively recapitulate the time-dependent and multi-stage progression of this cancer because it happens over a period of time. But the disadvantages, again, is that cannot be used for all tumor types because all cancers are not chemically induced. The time frame to introduce these mutations in these models are prolonged. So you have a longer time of animal maintenance, of having someone to take care of your mouse model. And again, you may have variable latency and penetrance for this disease. A little bit about this particular chemically induced model, because it's so widely studied, is that you introduce, so this is the initiation phase, and I'm showing you for squamous cell carcinoma. So you have a sub-carcinogenic mutagen, which you apply in the initial stages. And this is usually applied for about 2 weeks, and it causes a slow mutation. And then you have this promoting agent, which is your TPA, that goes on for about 10 to 40 weeks. And this causes an increased DNA synthesis inflammation an increased accumulation of mutation. And upon progression, you go from a normal skin to hyperplastic epidermis to papilloma to squamous cell carcinoma. So this is a slow progression to having your cancer generation. And it's very useful to study these chemically induced models in a cancer that's relevant to this type. And so because of that, there are many chemically induced models that have been studied over the years. For skin cancer, the model that I just described. For lung, you can have it induced with nitrosamines. With liver cancer, breast, colon, and bladder, you can have chemical induced mouse models. You can have viral infection models, and there have been many successfully studied models, and several have won Nobel Prizes. As you can see here, the Rau-sarcoma virus was initially discovered in chickens in 1911. Then we had the mouse mammary tumor virus, where you had virus infection models or virus-induced cancer models. The other models that have been studied subsequently have been in RAF, FOS, CFMS. These are engineered viruses that can now be used routinely in the labs to induce genetic changes that lead to cancer. But again, you can only study it in a subtype of cancer where such model is relevant. Next, I'll go into a very specific type of models that we use extensively to study any type of cancer is a xenograft model. So there can be broadly three types of xenograft. Xenograft that is developed from a cell line, and it could be a human-derived cell line or a mouse-derived cell line, or it could be a patient-derived xenograft, which is a tissue that is derived from patients through biopsy or surgical resection, and then circulating tumor cell-derived xenograft models. So what is the difference between having a patient-derived xenograft model versus a genetically engineered mouse models? So genetically engineered mouse model has a native vasculature, which is developed in the mouse. So these are mouse vasculatures. It has an immunocompetent background, so it has an innate immune system that's intact. It can be orthotopic tumor growth, so a lung cancer model is seen in lung cancer, and there are benefits to that because you can study a tumor in its own microenvironment, but it has very limited intratumoral heterogeneity, and it does not recapitulate what you see in patients 100%. But what you see in PDX is it is a murine vasculature again. It has severely limited immune system because you cannot take a human tissue and put it in an immunocompetent mouse model because there will be a graft rejection. So you have to put it in severely immune deficient mice. And that is a problem because most of the immunotherapy studies cannot be done in a PDX model. It has an admixed murine and human stroma. It is mostly orthotopic, or it can be ectopic as well. I'll talk about both. And it has a full range of molecular subtypes because you can recapitulate what you see in patients in terms of tumor heterogeneity. So in a transplantation model, as I said, it can be ectopic. Ectopic means it is subcutaneously implanted, like the mouse will have a flank tumor that is subcutaneous, or it can be orthotopic, like it is implanted into the organ of your choice. Like if it's a lung cancer cell, it is implanted into the lung of the mouse. So that's called orthotopic. The advantages of having a cell line based xenograft is that it is very easy to maintain cell line indefinitely in the lab. So it is very easy to maintain your population of that cell line. It's very fast to generate large number of xenograft tumors because of your abundance of cell lines that you can get. It's relatively homogeneous of tumors, and it makes it easy to detect differences. And it's comparatively cheaper than making a genetically engineered mouse model. The disadvantages, and the biggest of that, is the lack of immune system, because now most of the studies you would want to have an intact immune system. So the lack of immune system is a major drawback here. And it is inaccurate in terms of histopathology compared to human tumors. And the evolution of tumor cells during culture. So if you culture tumor cells for a long time in the lab, they're bound to have certain evolution, which may not completely mimic what happens in patients. So these are sort of the drawbacks of having a cell line-based xenograft model. The other that I was talking about is cell line-based allograft. So what I mean by allograft is taking cell from a mouse and putting it into a mouse. So that is an allograft. The advantages are it is very easy to, again, maintain these murine cell lines indefinitely. It is very fast to generate these xenograft models. It's relatively homogeneous tumors that you can study. And the biggest, I think, advantage is that since it's mouse to mouse, you can study that in an intact immune system. The disadvantage is that it's a mouse cell rather than a human cell, so it might not recapitulate whatever you see in a human cell line. Again, it's an inaccurate histopathology because it's a mouse tumor. It's selective for cells. It gets adapted to plastic when you grow it in the lab, and evolution of cells during culture. And historically, it has a poor ability to predict responses of therapy to humans. Again, because of having a limitations of being a cell line derived model. So now what we do extensively is derive patient-derived xenograft models. So you consent your patients when they come into the clinic, and you either take tissues when you surgically remove the tumors. You can take blood or fluids, or you can even, after the death of the patients, you can have autopsy-derived samples that you can then implant in a immunocompromised mouse model, the NSG mouse, and those can develop tumors. The beauty of that is it recapitulates the complex genotypes and intratumoral heterogeneity of the tumors when you have a mouse-derived model, when you have a patient-derived model, sorry. What I will spend like a few minutes is, this is a new technology that people are using specifically for cancers where resection is a problem, is to get liquid-based biopsy or blood from patients and deriving animal models from there for biomarker discovery. And this is a xenograft, which is from liquid biopsy to a xenograft model. As you can see here, so the blood is collected from patients, and then we isolate the circulating tumor cells from these blood that is derived from patients. And they are injected into the flank of these mice. As you can see here, these mice develop tumors very beautifully. The beauty of that is, and I'm giving you specific examples, this is a lung model, small cell lung cancer model, is the metastasis that you see in patients, like the liver metastasis that developed in patient, also developed in the mouse. And if you have lung mets in the patients, you also have lung mets in the mouse. The biomarkers that you see in patients, like overexpression of NCAM or chromogranin, you see that in the mouse model as well. Even when you do a genomic study, the mutation that you see in patients is actually very faithfully recapitulated in mouse. So I think the take home from this slide is these mouse models actually very faithfully recapitulate what you see in patients, from having the same metastasis origins, from having the same biomarkers, and also the same mutations. I'll spend the rest of my talk talking about genetically engineered mouse models, as they are one of the most studied mouse models in cancer biology. Before I go into details, I'd like to sort of get you acquainted with a few of the terms that I will be using. So a non-germline GEMM is a mouse model that carries genetic modifications in some of its somatic cells, but not in the germline cells. Each model has to be individually generated, for example, transplantation or by injection. Whereas a germline model is a mouse model that carries genetic modification in its germline, and it's maintained through breeding. So the model does not have to be individually generated by transplantation or injection. Since the mutation is carried through generation, you can maintain this mouse model through breeding. A chimeric mouse model is a mouse model that has been generated through embryonic stem cell manipulation, and I will go into that in my talk. followed by injections of these cells into pre-implantation embryos. Again, I'll go details into the process that is followed to do that. And the resulting chimeric animals, the resulting animals is a chimeric animal. Conditional GEM, again, I'll go into conditional GEM in details, is a model that acquires an activation or inactivation of the original genetic modification that you see in somatic cells. And you can do that through homologous recombination through the Cre-LoxP system. And as I said, I'll go details into that. And there are inducible mouse models, like you want to turn on and turn off a gene according to when and where you want to. So for these, the inducible mouse models are important, and I will go into that, the TET response elements and how those can be used in a TET on, TET off system. So as I said, genetically engineered mouse models is widely used in basic cancer research and in translational oncology. It can be used to study cancer gene validation, cancer gene collaboration, tumor-host interaction, cells of origin, genotype-phenotype correlation, to study drug resistance, drug tolerance, anything, you name it, you can use these for your basic cancer research in the lab. For translational oncology, it has been proved to be very, very effective in studying biomarkers, in studying the effect of therapies like targeted therapy, immunotherapy, and the beauty of having a GEM model or genetically engineered mouse model is that they're immunocompetent. So you can actually study the effect of immunotherapy and other targeted therapies in this, additional cycles, and then combination therapy. So sort of GEM has found its place in every area of cancer research, and that's why they're so important to a cancer researcher right now. So what are the advantages and disadvantages of a genetically engineered mouse model? And as I said, every model will have its own limitation, but this is just an overview. You can do a stepwise progression through neoplastic stages. So it gives you the ability to introduce your mutation through stages. You can engineer what your specific mutation is going to be and when you're going to introduce that to study gene function and or reporters. It is very well established and understood technology. So now a lot of the labs have developed their own mouse models. So it's a very well established and understood technology. It's amendable. It's a very powerful genetic screening approaches that can be done through a genetically engineered mouse model. Tumors develop in the presence of an intact immune system. Again, that's a very big benefit. And you can study both neoplastic components and the stromal cells when you're doing genetically engineered mouse models. The biggest disadvantage, and that's the limitation for every mouse model, is that mice are not people. So mouse cells are not human cells. So what you see in the mouse may not be 100% recapitulated in clinical trials, and that's an inherent drawback of any animal model, actually. And the laboratory stains, they fail to represent the genetic diversity of human population because they are grown and bred in captivity, so they have a very homogeneous genetic makeup, so they do not recapitulate the genetic diversity that you see in a human population. The mouse tumors, they typically grow very fast, and that's one of the advantages too why we study mouse tumors, is because you can have your studies done in a relatively shorter period of time. But since they develop faster than what you see in patients, you do not see that whole time lapse of what you would see in a human tumor. It is complicated engineering strategy, so you may have to have like a dedicated personnel and animal facility for you to have a genetic mouse model. It is slow and expensive, so all labs may not be able to have such an advantage of having these mouse models in-house, and it requires a dedicated infrastructure. So having or doing research in a place like Memorial Sloan Kettering, of course, has its benefit because the infrastructure is very conducive to doing any sophisticated mouse model study. But it may be a drawback if you're not in a place that has these sophisticated systems. So I'll be talking about germline GEMMS in details because they have been used a lot in studying cancer biology. So the methods of creating a transgenic animals, the first is through microinjections. And I have a video for you, but I'll just quickly explain what microinjection means. Is that you transform or you genetically engineer a fertilized egg, and you harvest this freshly fertilized egg. You inject this fertilized egg into the male pronucleus with the DNA that has been manipulated with your target gene. And the pronucleus is fused to form the diploid zygote nucleus. So this is the video. Let me see if it works.
You may need to share.
Yeah.
Yeah.
So as you can see here, you're injecting your eggs into the pronucleus. And this is what microinjection is. I'll do it one more time. So you're basically introducing these eggs into a male pronucleus. Sorry. Sorry about that. So the next is the embryonic stem cell mediated transfer and the isolation. So you isolate pluripotent stem cells and have a desired gene that is inserted into these cells. And you have recombinant cells that are incorporated into the host's embryo. So I'll show you a graphical representation of this. So this is what you call a mouse blastocyst. And you have these embryonic stem cells that you have isolated from the mouse blastocyst. And you introduce your targeting vector or your gene of choice, either by electroporation or through other techniques, into your embryonic stem cells. And these rare cells that carry the target genes are then selected. And you now have your pure population of these pluripotent means they can develop all tissues of the body. So you have these pluripotent embryonic stem cells. You introduce these pluripotent embryonic stem cells into your blastocyst of your target. So you have what you call, so this is the outer layer is the trophoderm, and this is the inner cell mass. So you inject your embryonic stem cells into the blastocyst of your recipient blastocyst. Now, these blastocysts are then implanted into your foster mothers. As you can see here, we have injected that into a foster mother, which then gives birth to what we called a chimeric mouse. So what you have here is a mixed population of mouse, hence the color differences here. And you have mating between your chimeric mouse and a normal mouse. And now, when you have this mating, you have 25% of the population that develops a mouse with your target gene of interest. So this is how you have embryonic stem cell transfer. So I'll just recapitulate because I went through this quickly, is you take out embryonic stem cells, which are the inner cell mass, from your donor mouse. So you have these mouse blastocysts, you take out the embryonic stem cells from the inner cell mass. you engineer these embryonic stem cells and you put together, you insert your gene of interest. And now you have these engineered embryonic stem cells that you inject into the blastocyst of your recipient mouse. And when they're implanted, this mouse then gives rise to a chimeric mouse, which when then you breed with a normal mouse, you get 25% of the population, which will have your gene of interest. And then you go through repeated rounds of breeding with that mouse. And the last is the retroviral mediated gene transfer. There are several constructs that have been used over the years. So you can have a cDNA transgenic construct. You can have a bacterial artificial chromosome transgenic construct. It demonstrates what happens when you overexpress a certain gene. This has been used extensively in genetic studies. And it is used to create tissue-specific gene regulatory elements for more complex strategies. The advantages of that, it is very easy to engineer these kind of retrovirus-mediated germline GEMMs, and it's comparatively rapid and cheap. But the disadvantage is that you can often have very random site integration. You may not be able to control the spatial and temporal integration of the gene of interest into these mouse models. There are several other much more sophisticated systems right now, like having a short hairpin RNA knockdowns or having a lentiviral transgenic. Again, the advantages for both the systems, it's very rapid way to learn what your gene does in a certain system. And it is, you can have a pretty robust system of knocking down or knocking in a gene through these mouse models. But the disadvantages are your penetrance may not be 100%. You never really get a complete loss of gene expression through a short hairpin RNA, which is now being corrected through CRISPR-mediated mouse models, that you can get a complete knockout of your gene of interest. And with lentiviral transgenics, you can have random integration. So it may be hard to control the random integrations with the lentiviral constructs. And it can have a multiple copy. So ideally, your mouse model should have your gene of interest or your target mutations in a very controlled environment. You should be able to insert it where you want and when you want. But in these particular mouse models, that may be a drawback there. So to overcome all the drawbacks that I mentioned before, there have been advanced genetic tools that enable the construction of very complex mouse models right now. And that has been achieved through having recombinases, enzymes that catalyze site-specific recombination in your cells. So to overcome all the drawbacks that I mentioned before, recombinases have been very important. And having inducible alleles, to have the ability to turn on or turn off a gene as and when you want, inducible alleles have become very important. And I will talk about a model to study that. So what does homologous recombination mean? And I think it's very important that you understand the concept of homologous recombination, which I will again go in my next slide into a further detail. So a targeted integration of a DNA into a specific site through the genome homologous recombination has been used to make these recombinase mouse models. The use or the advantage is that it is a very precisely engineered gene loss, gene editing, or subtle mutations that can be studied, and you know where your mutations are, and you know when and where you have introduced that. The disadvantage is that the target mutations, if it's very important to the body, it can cause embryonic lethal phenotypes. So your pups may never be born, or they may die in utero. And they are slow and more difficult to carry out, and they are complex, as I will go more into the processes. So the advantage is that it's very precise, but the disadvantage is that it's slow and a difficult experiment to begin with. So what is homologous recombination? Homologous recombination exists in your body. It's a method of repairing double-stranded DNA breaks in your body. And the reason I want to talk about this in detail, just not in the context of mouse model, is because this is an important concept to understand before you even start designing your mouse model. So when you have double-stranded DNA breaks, which can happen with any assault that happens to your DNA, you will see these cuts. So what then happens is that you have these, what we call 3' overhangs that is created by the DNA before they start even repairing their DNA. This red right here is your homologous chromosome. Homologous chromosome is a chromosome that has identical or similar sequences to the DNA that has been broken, that has undergone double-stranded DNA breaks. So now when there is a double-stranded DNA break like this and you have these 3' overhangs, now what it does is it goes and invades this damaged DNA, it goes and invades your homologous chromosome because it needs to copy the sequence from the sequence of the homologous chromosome. So then what starts is called the repair synthesis or branch migration, where this damaged DNA is starting to copy the sequences that you see in the intact homologous chromosome. So it just copies and pastes what it sees in the intact chromosome, and then it makes a copy of that intact chromosome. So what you have here, right, in this black chromosome right here, is that you have a copy of the intact, this red chromosome here. So this is the process of homologous recombination, which happens all the times in your cells, which undergoes a double-stranded DNA breaks. It goes and copies from a homologous chromosome. So that has been used now to design mouse models. And I will talk about the Cre-LoxP system. So Cre recombinase is a site-specific enzyme that catalyzes recombination between two LoxP sites. So it is an enzyme that will cause the process of homologous recombination that I mentioned before when it recognizes two LoxP sites. It was first discovered in bacteriophages. What is a LoxP site? So this is essentially what a LoxP site is. It is a 34 base pair DNA sequence, which is a palindromic sequence on both sides, but it has this 8 base pair core sequence in the middle, which decides the directionality of this LoxP site. You can cause gene deletion, gene inversion, and gene translocation by using this Cre-LoxP system. So when a LoxP site is facing in the same direction and you have your gene of interest in the middle, when your Cre is in the system, you can cause a gene excision. So if your LoxP site faces in the same direction, you have what is called a gene excision. When your LoxP site faces towards each other, like you have these gene of interest in the middle and your LoxP site faces each other, now when you have Cre recombinase in the system, this is what you call a gene inversion. And it can also call translocation between two chromosomes, is if you have two LoxP sites, similar LoxP sites between two chromosomes, and then you introduce your enzyme Cre, then what you call is a reciprocation translocation. There is a strand exchange between these two chromosomes. There have been many mouse models that have been created using the Cre recombinase system, and you can look into these references. There has been many more created over the years. And I would also like to talk about something called a flex switch, which is a much more advanced system using the Cre recombinase. So what you see here is that what I mentioned, when you have two LoxP sites that faces each other, you have gene inversion. And gene inversion is usually reversible, like it switches between each other, which may be a problem when you're creating a mouse model if your gene of interest is reversible, like it switches back and forth. To overcome that, scientists have now come up with something called a flex switch. So you have two LoxP sites within which you have your gene of interest. So these are these purple and the green, which are similar to each other. So now when you have a Cre in your system, you first cause an inversion. So as you can see here, now you have caused the gene to invert. What you now have here is two LoxP sites that are in the same direction. So now when you have a Cre recombinase, it can actually excise the LoxP sites. So what you now have is two different LoxP sites between your flanking your gene of interest. So now this gene inversion becomes irreversible because a Cre can only recognize two similar LoxP sites. And it can be used extensively in designing or causing your gene editing system. The Cre considerations that you have to take is that it can produce Cre toxicity, it can cause side effects, some target genes may not recombine. So there are several advantages and disadvantages of having this Cre-Lox system. But the beauty is that you can introduce your gene of interest or the mutations where you want and when you want it. There are other systems that I would not go into the details that have also been used to study these conditional mouse models is the FLIP FRT. It's exactly the same. It's the FRT site instead of the LoxP site, and your FLIP is your recombinase. Again, if they're facing, the FRT site is facing the same direction, you cause an excision. If it's facing towards each other, it is causing an inversion. And if it's on two different chromosomes, it is causing a transversion. And now, again, if you want to build a more sophisticated mouse model, you can combine the Cre-LoxP and the FLIP FRT sites. The last model that I will be talking about is a TET-on system, because you want to have inducible system where you can introduce your, you can turn on or turn off your gene of interest as and when you want it. So the uses are you can have a dose-dependent expression of your gene products through administration of tetracycline, and this is the system that I'm going to talk about. The advantages, of course, of very precise temporal and amplitude control. And the disadvantage is, again, it's non-physiological gene expression. Again, this was first identified in a bacteria. So bacteria have these antibiotic-resistant genes. So here I'll be talking about tetracycline-resistant gene that is under a TET promoter. So when you have tetracycline in your system, this gene gets turned on. When you do not have tetracycline in your system, or you need to turn off, you have something called the TET repressor gene, which then goes and turns off the gene expression. So scientists have used that to now design their mouse models. So what you have is your target gene. It can be any gene of interest under a minimal promoter, and it will be under the TET responsive elements. This is the sequence of the TET responsive element. So in a TET-on system, you would have something called the reverse TET responsive transcription factor, which tetracycline gets bound to. So in a TET-on system, when there is tetracycline in your system, this tetracycline, this RTTA, will go and bind to your minimal promoter and will turn on the gene expression of your gene of interest. So essentially, with tetracycline in your system, your target gene gets turned on. In a TET-off system, it's completely different, because now you have something called the TTA transcription factor, which is, again, under the TET response elements. You have your target gene and your minimal promoter. Now, when you have tetracycline in your system, this TTA goes and binds to the TET response element, and it does not let it go and bind to the minimal promoter. So it actually, when you have tetracycline now in your system, it stops the gene expression of your target gene. So you can either turn on or turn off your gene expression by either using a TET-on or a TET-off system. It's a much more complex system, and I don't have enough time. And as I said, I'm happy to answer questions either via email or through via Twitter about going into the intricacies of these models. But all you have to remember is, if you want to turn on or turn off your gene of interest at a certain period of time, a TET-on or TET-off system can work beautifully by having tetracycline in your media or not. And there are other with doxycycline now with a dox-on, dox-off system, but it essentially follows the same principle of having an inducible model. So I'll just recapitulate the disadvantages of having a genetically engineered mouse model is that it relies on the expression of a single potent oncogene, and it may not recapitulate the complexity of a cancer patient, where there are much more genetic alterations that we can build a mouse model for. Although it's histologically and genetically similar to human cancers, you have to understand that any model, any animal model is limited by not having the complexity of a human body. It's a quantitative assessment. Sometimes you can have a problem with studying the downstream analysis, like, assessing your tumor growth or assessing your gene function may be a problem, because you may need much more sophisticated reporter elements and measuring criteria for studying these tumors over time. But nevertheless, it has been really important to study cancer gene and drug systems by using genetically engineered mouse models. And more recently, we have used humanized mouse models. If you remember, I was talking about patient-derived xenograft models, and the main disadvantage of having that was you did not have an intact immune system. So you could not study the effect of immunotherapy. So the newer technology we now have is that you have this immunodeficient mouse model, which is exposed to myeloablation and reconstituted with human stem cells. So you introduce CD35 positive human hematopoietic stem cells that makes a humanized mouse model now. And these promising models have been used in several high-impact studies now to study effect of immunotherapy in a PDX model. So I think that's where it has found its most benefit, is now you can take a patient-derived xenograft model, which is grown in immunodeficient mice, you can reconstitute the human immune system and study the effect of immunotherapy for that particular model. Again, this is an expensive system. You need very sophisticated system to study it. The penetration may not be 100%. So like any other model in cancer research, this also is riddled with its own advantages and disadvantages. In the summary, each model has its inherent limitations. So please choose your model very carefully when you're studying a particular disease and the question that you're trying to answer. Your question may be very simple, and you may not have to go into very complex mouse models to study them. You can get away with doing a cell line xenograft or a patient-derived xenograft, and you may not have to make a genetically engineered mouse model. So think very carefully about the questions you're asking in your research and design your mouse models accordingly. With that, I'd like to thank everyone for their attention, and I'd like to take any questions if you may have. Thank you so much.
Thank you so much, Dr. Sen, for such a great talk. So I'll go ahead and stop the screen sharing. Okay, so a couple of questions we have. The first is from Aaron Benoit. He asks, what is the difference between analogous and homologous tumors?
So analogous system is, I'll go back to my, sorry. So an analogous process is usually a process having a similar function. So if you want to study genes with similar functions, then you would choose your animal model that studies genes having similar functions, like a tumor suppressor or a gene causing cancer. A homologous process is a gene having a similar genetic sequence that are having a same embryonic origin or same genetic sequence to begin with. So it depends upon, again, whether your cancer is similar in function or it's similar in the genetic makeup that you're studying. And that's what differentiates an analogous versus a homologous process.
Okay, great. Thank you. Catherine Lackman asks, what kind of problems can come as a result of the gene being reversible with the LoxP site?
So the reversible is, so when you reverse your gene, so you're trying to study the function of a gene inversion. With the LoxP site, your gene inversion is reversible. So which means is you do not have any control whether you're studying the gene in the sequence of that you want to study or in the inverted sequence. So the fact that you want a mouse model is because you want to have the control over the exact gene sequence that you want to study. Let's say you're studying P53 or you're studying a particular gene of interest and you want to invert the gene and see its function. If you do not know whether the gene is going through a spontaneous process of inversion, so you do not know the function that you're seeing is in what process, is it through the inverted gene or is it not through that process? So that's why the flex switch is important, is because once you invert a gene and you keep it inverted, you would like to study what happens once the gene is inverted. So that's one of the limitations of having a gene inversion, which is reversible, is because you do not have control over the sequence that you're studying. And that's why the flex switches become very important.
Okay, thank you. The next question we have is from Claudia Rodriguez. She asks, how often do doctors and scientists use animals to help cancer research and conducting cancer research?
Well, now, very often, like all research is sort of, we try to do it in cell lines and we try to do it in the mouse model. Because as I said, with the mouse model, you're getting one step closer to studying the human biology, right? So every study that we do now, I think every or majority that the studies that we do now, we want to do it in a mouse model, because especially when you're doing a drug study, you can do in vitro studies. You can see the effect of the drug on the cell lines and see what kind of effect that drug might have on the system. But once you want to see what happens to a tumor growth once you treat that tumor, and that cannot be studied in a cell line. So to study the effect of a drug, to see the toxicity of this drug, you have to go into a mouse model. And also to see the effect of a gene, again, you have to go into a mouse model. So I would say majority of the times now we use animal models in cancer research.
Thank you. Ashwar Patri asks, is the inducible mouse model similar to a knockout mouse?
So yes, so a knockout mouse is you're knocking out your gene of interest. Inducible mouse model is, again, you're knocking out the gene of interest, but you're doing it in an inducible manner. So you are going to knock in and out or turn off and turn on a gene with your, so in a knockout model, you've just knocked out the gene. So you do not have the ability to turn that gene on if you want to. So you've knocked it out, it's no longer in the system. In an inducible mouse model, you're actually turning on or turning off a gene, depending upon the system that you're using. So it's repressing the expression of the gene and turning on the repressed expression of the gene. It's like you've removed the bulb from a lamp. That's knockout. Or you're turning on and off the switch to turn on the bulb or off the bulb according to when you want the light or not. So that's the difference between a knockout and an inducible mouse model.
Okay, great. We do have another question from an attendee. They asked, how do labs handle the ethical side of using mouse models and other animal models? For instance, how much to feed them, housing in cages, etc.
Oh, we go through severe ethical considerations. Like every mouse model that we design, we study, we consider everything. So we consider whether you at all need mouse model for that study. So that's your first consideration. If your study can be successfully done in a cell line, we never use a mouse model. The second consideration is, does your facility provide a humane system for even growing or breeding or keeping these mouse models? Second is, how are you keeping them? How are you euthanizing them? So we take every measure possible. There are committees at MSKCC or at every institute that has an animal house. There are committees that are very stringent about overseeing that a mouse study is being done in an as humanely manner as possible. You cannot leave your mouse alone. I mean, there are vets that take very good care of our mouse. So I guess a short answer would be we take really good care of these mouse models because they are absolutely essential for our studies, and also we want to do it in a very humane manner. So we would never sacrifice or use more mouse in our experiments than required, and we will treat them or sacrifice them in as humanely manner as possible.
Okay, great. Thank you so much, Dr. Sen, for such an informative talk. And the remainder of the questions, we will send them to Dr. Sen, and she will respond to them via Twitter. Again, we will be emailing you our survey to provide feedback, and we'll announce registration for our PI Spotlight seminars tomorrow. Thank you again, Dr. Sen, for your time. We greatly appreciate it.
Thank you so much. And I really appreciate the questions. As Vicky mentioned, I am happy to, I see a lot of questions that I really want to answer, like what is a neoplasm, what schools I went to. So I'm happy to answer all your questions via email or Twitter. Please send them to us.
Yeah, we'll be sending you a copy of all the questions we weren't able to answer.
Thank you.
Thank you, and have a good rest of the day, everyone.